So picking up where we left off, we were talking about chemistry and some of the basic elements, such as what defines an element and some of the chemical properties of water. Now we're going to move on to some other very important molecules, and the first of these being carbon. Carbon is very important because it has 4 electrons in its valence shell, and it needs 4 more to attain a stable octet, and thus it can form 4 covalent bonds, and covalent bonds are very strong. And so it can form a number of bonds with a number of different elements, and it can also make these long chains of carbons attached to other carbons, and those are called hydrocarbons. So it is unique in that way, and because of its unique properties, it is a building block. And so most of your molecules in your body are made of carbon. And we'll talk more about them when we talk about the other macromolecules. Okay. Now we talked about different types of elements, ions, isotopes. There are also different types of compounds. Compounds that are different are called isomers. And isomers have the same molecular formula, so the same number of atoms, the same kind of atoms, but they differ in a few ways. They differ in structure, and those are called structural isomers. Okay, they can also differ in attachment. So, again, they can have all the same elements in their compounds, but just certain compounds are in different attachments, attached to different carbons, so on and so forth. And we'll give a couple examples of these in a minute. One type of stereoisomer is called an enantiomer. And these are molecules that are mirror images of each other. So if you look at these two molecules in our figure here, they look exactly the same, the only difference being they are mirror images of each other. So it'd be like a right hand and a left hand. They look the same, but the thumb is on the opposite side. Okay, same thing with the molecules. They are composed of the same elements in the same configuration, but the Y, okay, is on the opposite side. Okay, and if a molecule is able to do this, not all elements can, or not all compounds can. It is called chiral. So this is, or it has a component of chirality. So macromolecules are all polymers, and polymers are made of repeating units called monomers. And the bonds are formed between monomers in a process called dehydration synthesis, and they are broken between them but in a process called hydrolysis. Carbohydrates there are 4 different types of macromolecules. Carbohydrates, nucleic acids, proteins, and lipids. Okay, so we'll start talking about carbohydrates. Now, they typically have a 1-to-2 ratio of carbon, hydrogen, and oxygen. So 2 hydrogens for every carbon and oxygen. The oxygen and carbon are in the same ratio. Typically, it doesn't always follow that, but it's typically or close to it. So you could write the chemical formula CH2O N, and N being the number of that monomer. Okay, so if you had 15 of those, then you would have C15, H30, and O15. So the bonds between the carbon molecules, and really the bonds between any compound, form or store energy, which is released when they are broken. Starch is carbon atoms that are bound together in plants. So we call those starches. The same thing happens in animals, but it's not called starch, it's called glycogen. Okay, a monosaccharide then is a 6-carbon sugar. There are 3 types: fructose, glucose, and galactose. There are other types as well, but these are the most common. And they are actually have the same chemical formula, C6H12O6. It follows that 1 to 2 to 1 ratio. But they are different in their structure and are different isomers of each other. So fructose and galactose are structural isomers. So the carbons are, the double-bound oxygen is on a different carbon. So structurally it's different, everything else is the same. Glucose and galactose are stereoisomers. So the OH and H are on opposite sides. That's the only difference between the two. Okay, disaccharides are 2 monosaccharides that are bound together. So for example, sucrose, which is table sugar, is a fructose and glucose bound together. Maltose is another sugar which is made of 2 glucose molecules. A polysaccharide then is made of 3 or more monosaccharides, and again, cellulose, which is found in plants, lignin, and chitin, which is found in arthropods, insects, also found in fungi. They just have a lot of these sugars bound together in long chains, or at least in chains that are 3 or more. Okay, again, the monomer of carbohydrates then for these saccharides, polysaccharides, is sugars. For your normal, or for your smaller molecules of carbohydrates, the monomer would be the carbon, hydrogen, oxygen repeating. Okay, so nucleic acids then are another type of polymer, and their monomer is called a nucleotide. And this structure right here to the right is a nucleotide. Okay, a nucleotide is made of a phosphate molecule. That's this in yellow. A sugar. That's this ring structure here. And then a nitrogenous base. So a base, if it's not an acid, it's a base, and it has lots of nitrogen in it. Okay, in DNA, this sugar is deoxyribose. Okay, deoxyribose is a 5-carbon sugar. Those are labeled here, 1, 2, 3, 4, 5. So you don't see the carbons. They aren't actually drawn in this chemical formula, but each of these angles is actually where a carbon atom sits. Okay, so it has 5 carbons. That is deoxyribose, and then ribose would be at the 2' carbon, it would have an H bound there. And deoxyribose would have an H, so DNA would have an H there, and RNA has ribose, and it would have an OH. And that's the only difference between the 2 molecules. DNA has a nitrogenous bases that are called adenine, thymine, guanine, and cytosine, and RNA has the same, except instead of thymine it has uracil. And so that's what these 5 bases are, and these form bonds between each other. Now the nucleic acids, or sorry, the nucleotides are made by binding nucleotides together to make a nucleic acid. This bond is called a phosphodiester bond. So it has a phosphorus in the middle, and then these 2 bonds where it binds to a carbon here and a carbon there, those are called ester bonds, and there's 2 of them, so it's a phosphodiester bond. Okay. DNA, as we all know, is the molecule of life that is able to be inherited from one generation to the next. So it stores information, and it does this by making a sequence of bases. So we said adenine, cytosine, guanine, and thymine were 4 bases of DNA. The sequence, so the order of these bases, is a way of storing information. And that information can be used to make new DNA molecules or to make proteins. It's arranged in a double helix structure. This allows it to be very compact. And the double helix isn't completely uniform. There are parts where the gap is longer and parts where the gap is shorter. But it is a double helix, meaning a double twisted structure. And what keeps it together, it's made of 2 strands of DNA, and they're bound together by hydrogen bonds that form between the different bases. So adenine and thymine bind together, cytosine and guanine bind together, and then if it's an RNA molecule, uracil binds to adenine. Alright, so RNA is the other type, and there are 3 types of RNA. Messenger RNA is made by enzymes that basically read the DNA and they match the bases with RNA bases. Okay, and this process is called transcription. It does not take a specific form, it's usually just a long string of nucleotides. Okay, ribosomal RNA or rRNA forms, again, it's also made by reading DNA, but then it forms a specific structure that is used to make proteins during the process of translation. tRNA is also used in translation to make proteins, and what it does is it carries amino acids to the ribosome, and then the ribosome, while reading messenger RNA, binds together amino acids. And we'll talk about amino acids in a second. There are a few other nucleotides. DNA and RNA are the ones we're probably going to spend the most time talking about. But there are a couple other ones. ATP is the molecule in the body that is used to store and release energy. And this is adenine triphosphate. And it's made of 3 phosphates, and what happens is when you break one of these phosphates off, it releases energy. NADH and FADH2 are also used to carry hydrogen atoms through the electron transport chain. We'll talk about that when we talk about metabolism and how the body creates energy. Alright, so proteins then are our third macromolecule. They are made from polymers of amino acids, and they have multiple functions. Basically, they are the workhorses of the body. They are the ones that are actually functioning to take care of chemical processes that happen within the cell, and also physical processes that are used in animals and plants and all life. They act as enzymes, so they help reactions go faster. So one example is catalase, which helps break down hydrogen peroxide, which is a reactive chemical, into a non-reactive chemicals, water and oxygen. They're used for defense, so our skin is made of protein called keratin, and that keeps organisms and pretty much everything out of our body. Also, our immune system is made of a bunch of proteins and antibodies that help fight and identify infected cells. They're also used for transport, so they will bind to things and then transport them through the bloodstream. Used for support, again, muscles, skin are made of protein and support other structures and bones in the body. Motion, muscles, of course, are how we move our skeleton, also how we move things through our digestive system. They regulate, one thing they do is regulate on the cell membrane, they will regulate what molecules come in and out of a cell, and they are also used for storage, so they can make little compartments where you can hold things within the cell as well. Okay, so those are the basic functions of proteins. The monomer of proteins is called amino acids. I just mentioned that before when we were talking about RNA. And the bond they make is called a peptide bond. The composition of amino acids is they have a central carbon, and as we talked about before, a carbon can make 4 covalent bonds. So this central carbon is going to make 4 covalent bonds. It's going to make a bond with a hydrogen atom. It's going to make a bond with a carboxyl group, which is C, a carbon, 2 oxygens, and a hydrogen. It's going to make a bond with an amino group, which is a nitrogen and 2 hydrogens. And then it's going to make a bond with an R group. And the R groups vary, and that's what's going to define what an amino acid is by the R group. Okay, the peptide bond is formed by a dehydration synthesis reaction between the amino and carboxyl group. So the amino group of one side and the carboxyl group of the other side, between 2 amino acids, they lose the water, and then that nitrogen and that carbon bind together. That's a peptide bond. Okay, these are amino acids. I'm just showing you this. You don't have to know all these. I'm just showing you this to show you that in white here, that is an R group. Okay, and that's what defines the difference. Other than that, they are all the same. But this R group has a CH3 on it. That's called alanine. This one has a long chain of carbon and hydrogens. That's leucine. There are some that are acidic or charged. There are also some that have certain structures off of them. This is called a benzene ring, okay, which is just a bunch of carbons that are double and singly bonded together. And there are some with a special function like methionine, which helps identify the start of a protein. So when a protein is made, there are 4 different levels of structure. And the first one is just the sequence of amino acids that are bound together. Okay, so you may have an alanine connected to a valine, connected to an isoleucine, and so on and so forth. Probably can be hundreds of amino acids long. And that sequence determines its function. Within that sequence, you will have an interaction between the R groups within the different amino acids. And that will make secondary structures, such as this beta-pleated sheet here, which is where hydrogen bonds are formed between different amino acids. Or they can make a helix structure called an alpha helix. Kind of like DNA, only rather than being a double helix, it's just a single helix. Okay, those are secondary structures. Tertiary structures are the final folded shape of an amino acid. So after you have the whole sequence and all the secondary structures together, they may have has a beta-plated sheet here, an alpha helix here, another one here, and so on and so forth, and creates all these structures. And then once it's all folded together, that's the tertiary structure. Now you may have multiple proteins actually interacting together as well, and that's going to make up the quaternary structure. Okay, so for example, hemoglobin we talked about is made of 4 globin molecules that are bound together to an iron-containing filament. So it's made of 4 interacting subunits, and that's what makes up its quaternary structure. Okay, each subunit is actually an individual protein that has their individual tertiary structure, but together they make a hemoglobin molecule. There are other important components, though, of a protein and making a protein and its shape. Chaperonins are usually needed to fold the sequence of amino acids into the right shape. And if they aren't folded correctly, then they will not function properly. So, for example, cystic fibrosis, the proteins are fine, the sequence is fine, everything is fine except the chaperones do not fold it correctly. Another process called denaturation happens when the protein is under conditions that create it to unfold. When it unfolds, it loses its shape and no longer functions. So if it's too hot, too cold, too acidic, too basic, then it will denature. Okay, our last macromolecule are lipids, and these are, we defined these before, these are hydrophobic molecules, meaning they do not like water, they do not form hydrogen bonds with water. And this is includes our fats, waxes, oils, and some vitamins as well. So vitamin D is a lipid. There's vitamin E. There's a bunch that are lipid-based. And also includes animal fats. So we have some bacon grease on the bottom left there. Vegetable oils, olive oil there on your right. Earwax, also beeswax, it's a lipid. And then testosterone is a hormone, which is also a lipid. It does not dissolve in water. Okay, lipids are made, well, one type of lipid is a fat, which is made of a glycerol. A glycerol is basically half a sugar. It's a 3-carbon molecule. And it binds to fatty acids. So Fa here is abbreviating fatty acid. If you have 3 of them bound to a glycerol, you can have 1 or you can have 2, but if you have 3 of them, it's called a triglyceride. And so people talk about these when they're talking about food. It's something that is high in triglycerides. It's usually not good for you. It's high in fat, high in lipids. Okay, the fatty acids are not all the same, and they differ in structure, and that's why lipids take on different shapes or different structures, I guess, in the same temperature. So an unsaturated fat is one that has double bonds. So you can see here this lipid, this triglyceride to our right, has a bunch of double bonds between the carbons, and that means it does not have as many hydrogens. Those double bonds actually make kinks in the fatty acids, so that's why this structure is all kinked here. Okay, these are better for you nutritionally. They are harder to break down. They aren't as efficient as energy, and then when compared to saturated fats, such as animal fats, and they don't have any double bonds. They're just long hydrocarbon chains, and they align neatly together. Okay, these, because they don't have any double bonds and they fit together so nicely, at room temperature they are solid in nature. So this would be like your bacon grease or your animal fats or your butter. Okay, whereas this would be vegetable oils, olive oil, and most of your oils are going to be unsaturated. So there are a few other types of lipids. One of them is called a phospholipid. These are very important for cellular structures. A phospholipid has a phosphorus head, which is hydrophilic, and a lipid tail, which is hydrophobic. And what will happen if you put these in solution is the hydrophilic part will point towards the water, and the hydrophobic part will point towards other hydrophobic parts. So that'll either make a lipid bilayer, or it will make this structure called a micelle. These are very important for digesting and transporting fats in the human body. Okay, the lipid bilayers are important for making cell membranes. And that's it for our macromolecules. If you have any questions, be sure and write them down, and we will go over more of this on Tuesday. Thank you.